Okay, now we're going to discuss the second type of cell division called meiosis. Meiosis is only used for making eggs or sperm, called gametes. These are genetically different from the original cell. So let's talk about human chromosomes for a minute. Humans have 23 pairs of what are called homologous chromosomes. Think about the word homo. What does homo mean? Homo means same. So homologous chromosomes are the same number chromosome. So we know humans have 46 chromosomes, but these occur in 23 pairs. Why do we have pairs of chromosomes instead of 46 different ones? Well, that's because we got one set of chromosomes from your mother, from an egg, and we got one set of chromosomes from your father, from a sperm cell. And when the sperm fertilized the egg, those chromosomes merged into one nucleus, and that's the 46 chromosomes that make up your body. So a pair of homologous chromosomes is located down here. Look at the picture on the right. So let's just say this is chromosome number 1, and then this is also chromosome number 1. On each of these chromosomes, you can see this colorful striping pattern. These are called locus. Those are the places of genes. In homologous chromosomes, they have the same genes all the way across. That doesn't mean that they code for the same color eyes, it just means that maybe they all code for eye color. So they're coding for the same characteristic. And they're exactly the same size and shape, centromeres are in the same spot, and these patterns of where the genes are located are exactly the same. We can take our 23 pairs of chromosomes and divide them into two types of chromosomes. We have autosomes, which we call body chromosomes. They're numbered 1 through 22 based on size. And our last pair of chromosomes are called sex chromosomes. And our sex chromosomes determine what biological sex we are. So you can either have an X and a Y, or you can have two X's. Those are the normal. Notice that I'm distinguishing sex from gender. Gender is an identity, whereas sex is a biological structure. So gametes are something called haploid. This means that they have half of the set of chromosomes, or one set, half of the total number of chromosomes. Eggs and sperm are the only gametes that we make. Another type of cell is called diploid. Diploid is all of the rest of your body cells that have two sets of chromosomes. So we call diploid cells 2N. N is the number of chromosomes in a set. For humans, N is equal to the number 23. So a diploid cell in humans would be 2 times 23, which is 46. So that's every cell in your body, your eyes, your nose, your stomach, your liver, your heart, except eggs or sperm. When we do meiosis, we make haploid cells called gametes. The only gametes are eggs and sperm in humans. Meiosis is actually going to take us two divisions. In mitosis, we divided once and made identical cells. But since we want to cut the number of chromosomes in half, we're going to divide twice. So we start with a cell in the beginning of interphase. Again, we're going to go through the same phases of interphase. Growth, synthesis. So synthesis is going to happen here, where we're going to take chromosomes that were single, and now they become multiplied. So when we enter meiosis, we have two copies of the entire genome. The first division is called meiosis 1. This is when those diploid cells are going to separate. All of our pairs of homologous chromosomes will separate, so that one chromosome goes one way, one chromosome goes the other way. Meiosis 2 is called the second division. This is where each of these chromosomes is now going to split and separate again. So by the end of meiosis, we will have divided twice, and we are going to make 4 haploid cells. So we're going to have 4 cells that have half of the number of chromosomes as the original cell. In this image, you can see that that works out. In the original cell, there are only 2 chromosomes. And in all of the daughter cells, each one only has 1. So that's half. This creates the human life cycle, this difference between gametes and adults. So humans in the ovary or testes, depending on sex, will go through meiosis to create eggs or sperm. Those eggs and sperm can then come together in a process called fertilization to create a zygote. A zygote is that first cell that you ever were. That zygote then goes through mitosis, which we already covered, to make new bodies with ovaries and testes that can again make gametes. So let's go through the phases of meiosis. Meiosis is split into 4 phases: prophase, metaphase, anaphase, and telophase. And we just use the Roman numeral 1 or the Roman numeral 2. So meiosis 1 is dominated by homologous chromosomes. The key characteristic of meiosis 1 is that the homologous chromosomes are what are moving around and separating. So in prophase 1, we see the same things that happened in prophase in mitosis. The nucleus is being broken down, the spindle fibers are forming and separating to opposite sides of the cell, the chromosomes have condensed. The only thing that's different here is that now we're looking at homologous sets of chromosomes. So they've kind of colored these, maybe blue came from dad, red came from mom, and so these are one set of homologous chromosomes. Look at what's happening at the legs here. So these 2 homologous chromosomes have small portions of the legs that are crossing over each other. Prophase 1 of meiosis 1 has a very special process that happens called crossing over. And this is when pieces of the chromosomes actually break off and switch. And this is going to create a lot of genetic diversity for the eggs and sperm. In meiosis 1, metaphase 1, the next step is that the homologous chromosomes line up along the center. So you can see how it's not all 6 of these chromosomes across the center, but they're lined up in pairs. This way, when we separate, each of the new cells gets a complete set of chromosomes. Anaphase 1 is when homologous chromosomes separate. Notice that the sister chromatids are still attached. We're going to separate those later. And in telophase 1, we have 2 separate nucleuses forming. These nuclei are now haploid. In the beginning, we had 6 chromosomes, and now each one is only going to have 3. Same things happen in telophase as in mitosis. The spindle fibers disappear, the nucleus reforms, the chromosomes get loose, and then the cell will split with cytokinesis. It's important to note that the cell is not going to do interphase again. It's going to go right into the next cell division. So the next cell division is called meiosis 2. Meiosis 2 is dominated by sister chromatids separating. So instead, meiosis 1 was talking about homologous chromosomes. Meiosis 2 is talking about the division of sister chromatids. So in prophase 2, we're going to see the nucleus disappear again. We see 2 centrosomes, and the spindle fibers starting to appear again, and the chromosomes are starting to condense again. In metaphase 2, all of the chromosomes line up along the metaphase plate, so the equator at the center of the cell. In anaphase 2, all of the chromosomes separate. So now we see the sister chromatids are separating. And then in telophase 2, we start to reform all of our 4 daughter cells. The chromosomes get loose, the spindle fibers disappear, and the nucleus starts to reform. Notice how we now are going to have 1, 2, 3, 4 daughter cells. Each daughter cell looks a little different. These ones have more red chromosomes, these ones have blue chromosomes. Look at the tips of some of these chromosomes. Some of them are different colored. That's a result of crossing over all the way back in prophase 1. Some pieces of the chromosomes broke off and switched. And so all 4 of these daughter cells are going to be genetically different. Okay, let's compare mitosis and meiosis. So mitosis makes identical exact clones of the mother cell. Meiosis is going to cut the chromosome number in half, so it's making genetically different cells. Mitosis only has 1 cell division, whereas meiosis has 2. Mitosis only makes 2 daughter cells, whereas meiosis makes twice as many, 4. Mitosis occurs in all of your body cells. Meiosis is only used to make egg and sperm. So the functions of mitosis are for cloning, asexual reproduction, growth, and tissue repair. Meiosis is only used for sexual reproduction. So we saw how it was important to cut the chromosome number in half. Think about what would happen if you didn't cut the chromosome number in half. The next generation would have twice as many chromosomes. Well, that wouldn't produce a viable offspring. So meiosis is really important for cutting chromosome numbers in half. Meiosis also has another function that's really important, and that is creating genetic diversity. Genetic diversity is really good for populations of species. Sometimes when species use not diverse genetically organisms, they can cause big problems. In Ireland, when the Irish had to immigrate to the United States, they mostly did so because of the Irish potato famine. The Irish were doing monoculturing, which means that they were only growing one single species of, potatoes. But the potatoes got infected by a mold that killed them, called the blight. And millions of people died and had to leave their homes because their one single crop died. If they had been growing a wide diversity of crops, it probably would not have had as big of an effect. There's an outbreak of Ebola in the Congo, and there was one in West Africa several years ago. Ebola is very virulent, but some people survive. So the population will go on to be full of people who have resistance to that virus. Think about what would happen to the population if everybody was susceptible, if we were all genetically the same. Nobody would live. It would decimate us. So generally, populations that have genetic diversity do better than populations that don't. So meiosis helps us increase the genetic diversity in humans. So look at the 2 pictures. This is showing what happens in metaphase, when the chromosomes line up in the center. If this cell only has 2 homologous pairs, right, 2 chromosomes from dad, 2 chromosomes from mom, they could line up like this, where both of dads are on one side and both of moms are on the other. Or they could line up like this, where the left side has 1 from dad and 1 from mom, and the right side has 1 from mom and 1 from dad. So there's 2 different possibilities for lining up during metaphase 1. This is called the independent orientation of chromosomes. What these 2 chromosomes do and what these 2 chromosomes do are completely independent. It could happen as possibility A or B, and it's random. Eventually, possibility A is going to give us 2 possible combinations of chromosomes in the gametes. And possibility B gives us two different combinations of possibilities in the gametes. So the number of possibilities of combinations of chromosomes and gametes increases as we increase the number of chromosomes. Humans have 23 pairs of homologous chromosomes, which 2 to the 23 makes more than 8 million possible combinations of chromosomes to make in 4 different sperm cells or an egg. And so this gives you a huge diversity in what each individual sperm cell looks like genetically. This is one of the reasons why two people who are siblings from the same parents can look very different, because they can get a different combination of the genes. The other thing that allows for genetic diversity during meiosis is crossing over. So crossing over is the exchange of non-sister chromatids during prophase 1 of meiosis. The things I need you to know about crossing over: it increases genetic diversity, and it happens during prophase 1. So in the picture on the right, you can see this pair of homologous chromosomes. The blue is from dad, the red is from mom. During prophase 1, pieces of the blue and pieces of the red legs overlap each other and make kind of like an X. Then those pieces of chromosomes break off and reattach to the other side. So now I have this chromosome that used to be all blue and it has parts of red. This other one used to be all red and now it has parts of blue. So this means at the very end, it can make 4 different parental chromosomes, right? 2 that look just like the parents and 2 that have recombined into new genetic combinations. So this is 4 different combinations. That can happen for every single chromosome, and then it can happen 8 million different ways. So having crossing over plus independent assortment allows for trillions of different possibilities of sperm and egg cells. Then we have to fertilization for making offspring. So we have another random chance again, which sperm and which egg are going to actually eventually meet and become an offspring. So meiosis is really good at mixing up the DNA of the parents and creating genetically diverse offspring.